Okay, so let's give this short video a watch.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so that should give you a really good overview of the process so far. But the next question we're going to ask is what happens once that action potential propagates down the muscle fiber? And the answer is going to be those thin and thick filaments are going to slide against each other, causing muscle contraction. And we're going to learn how here in just a minute. Okay, so we have a couple of questions here. First question is, what does paramecium surround? Well, if we go back to our diagram of muscle, you can see that paramecium surrounds the individual fascicles. Okay, remember that a fascicle is made up of a bunch of individual muscle cells. Okay, makes one fascicle. This is one fascicle. This is one fascicle. And surrounding those fascicles is going to be the paramecium. And in that paramecium is going to be your artery, your vein, and your nerve supply, okay? Next question, what proteins are found in thin filaments? Well, recall that the thin filament is the actin filament, but it also has a couple other proteins associated with it. Here we have actin. It's going to be the string of pearls. This is actually 2 actin molecules intertwined. So actin's going to be that string of pearls. Tropomyosin is this long brown protein going along that's covering up the myosin binding sites on the actin molecules. And then you have troponin, which is associated with tropomyosin. And what happens, we'll see in a minute, is that calcium is going to bind to troponin, causing that protein to undergo a conformational change. And remember that that's just a change of shape. And when that happens, it pulls tropomyosin out of the way of those myosin binding sites. And the myosin heads on the thick filament are then able to attach here and pull the actin filament towards the center of the sarcomere. And I'll show you that in a sec. Okay, how many motor neurons and how many muscle fibers are contained within a large motor unit? Well, going back here just a minute. Remember that small motor units have less than 5 muscle fibers, but the large motor units are going to have thousands of muscle fibers. And remember that each motor unit only has one neuron associated with it. That's what makes it a motor unit. Okay, so this diagram is just an overview of the events in skeletal muscle contraction. We've covered number 1 so far. Neuromuscular junction, the excitation of a skeletal muscle fiber. So we've talked about the release of neurotransmitter acetylcholine from synaptic vesicles, right? That influx of calcium triggers the release of synaptic vessels containing acetylcholine. The acetylcholine then crosses the synaptic cleft and binds to acetylcholine receptors, which is then going to open and allow sodium into the cell and potassium out of the cell. And there's going to be a change in membrane potential here. That's step 1. Step 2, that change in membrane potential is going to move along the sarcolemma, right, along the plasma membrane of the muscle cell. And it's going to move down these T tubules, which is just an invagination of the sarcolemma, and basically excite every single one of these myofibrils, okay? Acetylcholine binding triggers propagation of an action potential along the sarcolemma and T tubules to the sarcoplasmic reticulum. Then the sarcoplasmic reticulum is going to release calcium. So we see calcium involved in the neuron terminal, and we see calcium involved here in the sarcoplasmic reticulum where it's released. Once calcium is released, we go on to step 3, and this is going to be in the sarcomere itself, cross-bridge cycling. And what happens, we just mentioned, calcium is going to bind to that troponin molecule, which is going to move that tropomyosin molecule out of the way. Myosin is then going to be able to bind directly to actin and slide those thin filaments past the thick filaments in the sarcomeres. The sarcomeres then are going to shorten, and the muscle itself as a whole is seen to contract. Okay, going forward, let's see, we covered all of this. Receptors, acetylcholine diffuses across the cleft, yep. Just seeing if there's anything else I want to say about this. No, we've covered that. Okay, let's talk a second about myasthenia gravis. This is going to be an autoimmune disease. You see it particularly in women, but it can happen to people of both sexes. This is going to be where antibodies bind to the acetylcholine receptors at the neuromuscular junctions and block the effects or the actions of acetylcholine there. These receptors, once the antibodies have bound, are going to be removed from the muscle fiber by endocytosis. And this is going to result in overall decreased muscle stimulation, right? Because there's fewer receptors at the cell surface to be stimulated. This leads to rapid fatigue and muscle weakness. And usually we see the eyes, the eyelids, and the facial muscles involved first. Can be followed by swallowing problems and limb weakness. In severe cases, if it affects your diaphragm, it can lead to respiratory failure. Most the treatment surrounds medication therapy as far as blocking those antibodies or trying to increase acetylcholine's action at the neuromuscular junction, okay? For example, if acetylcholinesterase breaks down acetylcholine, then maybe you would want to block the action of acetylcholinesterase so that you had more acetylcholine hanging around in a synaptic cleft. Okay, and that's kind of the concept behind an acetylcholinesterase inhibitor. Okay, so excitation, contraction, coupling. This is where stimulation of the skeletal muscle is coupled with the sliding of the filaments. Excitation and contraction coupling is going to include the change in membrane potential that occurs here at the end plate, the motor end plate. The action potential, or the impulse that travels from that change in potential, travels down the sarcolemma, and then all the way to the release of calcium from the sarcoplasmic reticulum. So here's that action potential traveling down the sarcolemma, travels down the T-tubule, and then the change in membrane potential is going to trigger release of calcium channels, release of calcium from the terminal cisterna of the sarcoplasmic reticulum. Calcium then just diffuses out of the cisternae and then into the sarcoplasm, where it's going to interact with troponin. Okay, so the end plate potential occurs on the skeletal muscle. So this is on the muscle side of the neuromuscular junction. It's going to be the minimum voltage change to trigger an action potential of muscle cells. Okay, so the threshold. Recall that acetylcholine receptors are chemically gated channels that open when acetylcholine binds to them, and this is going to stimulate sodium to enter the cell. So if you've got positive charges coming into the cell where the cell is negative on the inside, it's going to make it more positive on the inside, right? So it makes the membrane potential less negative. It goes from about negative 90 millivolts to negative 65 millivolts. That negative 65 millivolts is going to be the threshold, or the minimum voltage change that triggers an action potential on the muscle cell. Right? We said that an action potential is a rapid rise or depolarization, and fall or repolarization in the charge of the membrane. So a rapid rise, that would be making it more positive. And if you're making the membrane more positive, you're depolarizing it. And fall, so that would be making the membrane more negative, and that would be repolarizing it, okay? The end plate potential is going to reach threshold by causing nearby voltage-gated sodium channels to open. We said sodium then diffuses into the cell through those voltage-gated channels. The cell depolarizes, that is, it becomes less negative, eventually becomes all the way to positive 30 millivolts during this step. Just after the sodium channels open, they're going to close. Okay, so they're only open for a very brief period of time. And what happens is voltage-gated potassium channels open. And potassium is going to diffuse slowly out of the cell, and it's going to diffuse so much that it eventually returns the cell to its negative 90 millivolts resting membrane potential. This wave of repolarization propagates down the sarcolemma and down the T tubules, just like the wave of depolarization did. All right, so this graph is showing us the membrane potential over time. And here are some important numbers. There's negative 90 millivolts, right? So this is going to be the resting membrane potential. So here's the muscle cell at rest. But then step 2 happens. The threshold is going to be reached when an end plate potential is produced. So we got sufficient sodium. So this is going to be around the time where the acetylcholine is binding, okay, causing that end plate potential. The end plate potential is going to reach negative 65 millivolts, and then depolarization is going to occur. And this is your depolarization right here, this step. This occurs as voltage-gated sodium channels on the sarcolemma open, and sodium enters rapidly. And it's going to reverse the polarity from negative all the way up here to positive 30 millivolts, okay? So that's your depolarization step. Then those sodium channels close, and the potassium channels open. And potassium is going to leak out of the cell, making it more negative on the inside, right? So we're going to see this membrane potential start dropping, getting down into the negatives. It actually overshoots a little bit, gets a little bit more negative than negative 90, but it comes to an equilibrium right around negative 90 millivolts. All right, talked about all of this. Okay, so going back, excitation contraction coupling goes up to the point where intracellular calcium is going to be released from the sarcoplasmic reticulum. And that is going to increase calcium on the inside of the cell, and calcium is going to bind troponin, and that's going to induce a conformational change in the troponin molecule, making it change shape, and it's going to move that tropomyosin out of the way of those myosin binding sites on the actin molecules. So here you can see we have the actin molecules, the string of pearls. Okay, we've got 2 of those intertwined around one another. And then you've got the tropomyosin chain, or proteins, excuse me, that normally cover up these myosin binding sites. But you can see here's troponin, and it has calcium bound to it, and it has changed its shape and moved that tropomyosin out of the way. So now there's sites for these myosin heads of the thick filament to bind to. That's going to be called cross-bridge cycling, and it's going to be 4 repeating steps, okay? Cross-bridge formation, power stroke, release of the myosin head, and the reset of the myosin head. So number 1, you have cross-bridge formation. All this means is myosin is attaching to actin. It's forming a cross-bridge between the thin filament and the thick filament, those myosin heads. Number 2 is the power stroke. This is where the myosin head pulls the thin filament toward the center of the sarcomere. ADP and inorganic phosphate are going to be released during this step. I would know this step. I believe there might be a test question coming up on this step. So know where ADP and inorganic phosphate are released. Number 3, the third step, is going to be release of the myosin head. What happens is ATP is going to bind to the myosin head, and that's actually going to cause it to release from actin, okay? So remember that. ATP in cross-bridge cycling causes myosin to release. And then the fourth step is the reset of the myosin head. ATP is going to hydrolyze into ADP, an inorganic phosphate, by an enzyme called a myosin ATPase. This is what provides the energy to cock the myosin head and get it ready for cross-bridge formation and, more importantly, the power stroke. Okay? So it's just these 4 steps over and over and over. Okay, here. So we have the steps diagrammed for us. So here's our first step. Is going to be cross-bridge formation, okay? And here's your myosin heads. These 2 guys right here. Here's the rest of the myosin molecules. Here's your thin filament. And you can see the myosin heads attached to the actin on the thin filament, forming a cross-bridge, right? So this is like a little bridge here between the thick filament and the thin filament. That's your cross-bridge formation. Next step is the power stroke, or the pull. This is where the myosin head is going to move toward the center of the sarcomere, pulling this thin filament along. This is the power stroke. ADP and inorganic phosphate are released during the power stroke. Next is the release of the myosin head from those myosin binding sites on actin. That's going to happen when ATP binds to the ATP binding site on the myosin head. And then finally, the myosin head is going to reset. So it goes from this position to this position. ATP is going to be split into ADP and inorganic phosphate by that myosin ATPase, and that is what resets the head. And then you go back, you're ready for another cross-bridge formation. Okay, cross-bridge cycling. Cycling is going to continue as long as calcium and ATP are present. This is going to result in the sarcomere shortening as the Z discs move closer together. And there's going to be narrowing or complete disappearance of the H zone and the I band. The thick and thin filaments are going to remain the same length, but they just slide past each other. And I'll show you what I mean right here. You can see at rest. You can see the H zone here, where there's just thick filament. You can see here the H zone, just thick filaments in this area, and the M line, of course. You can see the I bands here that are just going to be primarily your thin filaments. Here are your I bands. What happens is when these thin filaments slide and basically are kind of arranged in a stack in between the thick filaments, you lose that H zone and that I band. All right? And you can see the Z discs here compared to here. The distance between the Z discs has shortened, and that is the actual muscle contraction right there. Okay, I have another video to show you here.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin,
which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T-tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Okay, so let's talk a second about tetanus and botulism. So tetanus, this is going to be a serious disease caused by a toxin that the bacterium Clostridium tetani produces. This toxin causes painful, prolonged muscle contractions that can paralyze the respiratory muscles, leading to death, okay? What happens essentially is the toxin travels to the spinal cord, where it blocks the ability of inhibitory neurons to send signals. So inhibitory neurons will send signals to relax the muscle. They will release, instead of acetylcholine, they'll release different neurotransmitters that have relaxing effects on muscle, okay? So if you block the ability to relax, you're going to have unopposed stimulation or unopposed input for contraction. And that's what happens in tetanus. You can prevent tetanus by getting vaccinated, which everybody should do, especially living in Texas, okay? Sorry, I'm sure you can hear my dogs snoring in the background. Next is botulism. This is going to be muscular paralysis caused by a toxin from the bacterium Clostridium botulinum. This, instead of preventing inhibitory signals, is going to prevent the actual release of acetylcholine at the synaptic knob. So no acetylcholine gets released. There's no signal sent to the cell to contract. Okay, so botulism causes a flaccid paralysis where there's not a contraction of the muscle. It's very useful as treatments for spasticity, such as that due to cerebral palsy, or I've seen it treated used in the treatment for migraine, chronic migraines. But it can also be used for cosmetic purposes, example, diminishing wrinkles. Botulism is the toxin is one of the most potent toxins, toxic compounds that we know of. But used in very minute doses, it can be beneficial therapeutically. Okay, so events in muscle relaxation. So what happens during muscle relaxation? You're going to have termination of the nerve signal. So the nerve impulse stops. So acetylcholine release is going to stop. When acetylcholine release stops, you're going to have no more action potential being generated on the sarcolemma. Calcium channels are going to close in the sarcoplasmic reticulum. Calcium that has built up in the sarcoplasm is going to be returned into the inside of the sarcoplasmic reticulum by calcium pumps. Troponin is going to return to its original shape, right, because calcium has diffused off of it. That's going to result in tropomyosin sliding back over those myosin binding sites on the actin molecules. And that's going to result in the thin filaments sliding back into their original place in relation to the thick filaments, and that's what causes sarcomere relaxation and the return of the muscle ultimately to its original position. Okay, I'm not going to go over these individually with you, but these questions are pretty crucial. I want you to understand each one of these, be able to explain them, because you're probably going to see some questions related to muscle contraction, specifically excitation, contraction coupling, and cross-bridge cycling on the test. So I want you guys to be well prepared. And if you have any questions at all, please shoot me an email. I'm happy to help. Okay, so how is energy supplied for all of this muscle contraction? Muscle cells themselves only have a little bit of ATP in storage. After about 5 seconds of intense exertion, all that stored ATP is going to be spent. Now, there's 3 different ways to generate ATP in skeletal muscle fiber. And remember, we're talking about skeletal muscle, not cardiac muscle, not smooth muscle. This is only skeletal muscle. There's an immediate supply via phosphate transfer. I'll show you that in a sec. There's a short-term supply via glycolysis, and go back to, chapter 3, where we learned essentially a whole semester of biochemistry in a week. And you will recall that glycolysis is kind of that first step in that oxidative pathway for glucose. So whenever you take in energy through food, the food's going to get broken down into its constituent parts. One of those constituent parts is glucose. Glucose is going to get transported inside of your cells into the cytoplasm to be turned into a total of gain of 2 ATP, okay? Net gain of 2 ATP. So it's not a lot of ATP, but it does help, and it is a short-term supply. Now, recall that the long-term supply is actually going to come from aerobic cellular respiration. That's where those byproducts of glycolysis, the pyruvate molecules, move on to the citric acid cycle, and then onto the electron transport chain, right, where oxygen accepts all of those electrons that have been shuttled through and have built that proton gradient that then cascades down its gradient, causing ATP synthase to produce ATP. Go back and review this. If this sounds all foreign to you, that's going to be a problem. You need to refresh yourself, refresh your memory on this. You don't need to know every single independent step at this point, but you need to be familiar with the big picture of glycolysis versus aerobic cellular respiration. Okay, because our muscles, the supply that they have, as far as how much oxygen supply they need, is going to depend on whether they tend to use a short-term supply or a long-term supply of ATP. So go back and review those mechanisms. Again, shoot me any emails if you have any questions whatsoever. Okay, so immediate supply of ATP. This is going to be phosphate transfer, okay? Transfer of inorganic phosphate. You can do it a couple of different ways. You can have two ADP molecules, and these can be combined with myokinase, the enzyme, helping out, and you get an ATP molecule and an AMP molecule at the end. But there's the short-term or immediate supply of ATP right there. Another way that another method muscles like to use is they like to stick phosphate on a creatine molecule and use the creatine molecule as basically a short-term storage site for phosphate. So they'll do that. You come in with creatine kinase, the enzyme, and that's going to cleave that phosphate off of there and produce ATP and creatine, okay? So that's immediate supply of ATP. Your short-term supply of ATP is going to be glycolysis. We said this is not going to require oxygen. Remember, this happens in the cytoplasm without or in the absence of oxygen. Glucose is converted into 2 pyruvate molecules. There's going to be 2 ATP released per glucose molecule. Finally is long-term supply of ATP. This is going to be aerobic cellular respiration. This requires oxygen, and it occurs within the mitochondria, right? So when we say mitochondria are the powerhouses of the cell, this is what we mean. We mean they make ATP, a lot of it, through aerobic cellular respiration. But they have to have oxygen. What happens is those 2 pyruvate molecules are going to get oxidized to carbon dioxide. They're going to transfer a lot of chemical energy to these intermediaries, NADH and FADH2. And then these guys go on to the electron transport chain and dump their electrons onto it. And those electrons supply energy to generate ATP, and that's going to be a net of 30 ATP, okay? Triglycerides can also come into this and be used as fuel to produce ATP as well. You can get more ATP from triglycerides that have longer fatty acid chains, okay? But big picture is that this long-term supply of ATP is going to require oxygen. How does your muscle get oxygen? It gets it through blood supply. Don't forget, though, that muscle also has myoglobin in it, which is an oxygen storage molecule. So muscle's pretty smart. It keeps oxygen on hand just for this purpose, for the long-term supply of ATP. It also keeps glycogen on hand for this purpose as well as the short-term supply. Remember that glycogen is the storage form of glucose. It's just a long branching chain of glucose molecules. So muscle cells store glucose store glycogen, and that glycogen is broken down to provide short-term supply of ATP and long-term supply of ATP. Okay, so here we have the short-term ATP versus long-term means of supplying ATP. So this is going to be glycolysis, all right, or the glycolytic pathway. You get glucose broken down into 2 pyruvate molecules, and that pyruvate then goes on to the citric acid cycle. And then all of these intermediate molecules that were made that are carrying electrons go to the electron transport chain and drop their electrons off with oxygen. All right. And build up that proton gradient between the membrane. And that gradient is the energy behind that gradient is what's used to power ATP synthetase and produce new molecules of ATP. So be familiar with this. You're going to see over and over again as you continue your career in science and in the medical field. You're going to see these metabolic pathways again and again. So get very familiar with them. Okay, so how which source of ATP gets used? Is it the immediate supply, the short-term supply, or the long-term supply? Well, that depends on the intensity and the duration of your exercise. So for a sprint that's like less than 10 seconds, most of your ATP is going to be supplied by that phosphate transfer system, okay, through those enzymes and these the conversion of 2 ADP molecules to an ATP and an AMP, and then the basically stealing that phosphate group off of creatine and making an ATP out of that. For a 400-meter sprint, so something around less than a minute, ATP is going to be supplied primarily by glycolysis after you burn up that immediate supply from the phosphate transfer system. Then you're going to be primarily using the breakdown of glucose in the cytoplasm or glycolysis as your primary supply of ATP. Now, once you get up to over a minute of exercise, you're going to shift to where ATP is going to be supplied primarily by the mitochondria. And this is what's going to depend on your oxygen supply to the muscles. Is going to be your biggest limiting factor here. All right, what does creatine kinase do? Remember that creatine kinase is going to be this enzyme here that can either add a phosphate to creatine or remove a phosphate from creatine. Mitochondria are abundant in muscle because of their role in metabolism. What is that role? Big picture here. Their role is to supply energy through aerobic cellular respiration. Okay, muscle fiber categorization. They're going to be categorized by 2 criteria. The type of contraction generated or the primary means used for supplying ATP. As far as the primary means used for supplying ATP, we have slow oxidative fibers, fast oxidative fibers, and fast glycolytic fibers. In slow oxidative fibers or type 1 fibers, contractions are going to be slow and less powerful. This is going to be for high endurance since ATP is supplied aerobically. So this is going to be for like your marathon runners. And these fibers are going to appear red due to large amounts of myoglobin, so a lot of oxygen storage in these muscle tissues. Next, you have fast oxidative fibers. These are type 2A or intermediate. The contractions are going to be fast and powerful, but they're going to primarily use aerobic respiration, but the delivery of oxygen is going to be lower than that in the slow oxidative fibers. They're intermediate in size and light red in color. And then there's fast glycolytic fibers, okay? Type 2B, fast anaerobic. These contractions are fast and powerful, but they're very brief. ATP production is going to be primarily anaerobic in these fibers, so largely through glycolysis. They are the largest in size, and they are white in color due to a lack of myoglobin. So I want you to focus on the difference between slow oxidative fibers and fast glycolytic fibers. Slow oxidative fibers are going to be for high endurance, such as athletes who run long distances. Fast glycolytic fibers, that's going to be for when you need something fast and powerful quickly. So, for example, a long jumper, somebody throwing a shot put, okay, discus, things like that. Okay, so a single muscle is going to contain a mixture of all of these fiber types, okay? Slow oxidative, fast oxidative, and fast glycolytic. But muscles are going to vary in their proportions of these fiber types in different muscle groups. So, for example, hand muscles are going to have a higher percentage of fast glycolytic fibers because your hands need to do quick, brief movements. Whereas your back muscles are going to have a higher percentage of slow oxidative fibers because you need to continually maintain postural support. Here's kind of the big difference I want you to take away from this. Slow oxidative fibers versus fast glycolytic fibers, especially for the test. You'll see long distance runners are going to have a higher proportion of slow oxidative fibers in their body just from conditioning. Okay, their body produces a higher proportion of slow oxidative fibers, whereas sprinters are going to have a higher percentage of fast glycolytic fibers. This is going to be determined primarily by genes, but also partially by training. So, again, the difference between slow oxidative fibers and fast glycolytic fibers, definitely know that. Okay, how do slow twitch fibers produce ATP? Going back right here, ATP is going to be supplied aerobically. So that's going to be through the electron transport chain or in the mitochondria. Which fiber contains the most myoglobin? We also said that's going to be slow oxidative, right? It needs the highest amount of oxygen supply or storage. And then which fiber type predominates in muscles used to maintain posture? That's also going to be slow oxidative fibers, okay? Muscle tone. This is resting tension in a muscle generated by involuntary nervous stimulation of the muscle. Some motor units are going to be stimulated randomly at any time, and then they kind of switch around, so they're changing continuously so one particular motor unit doesn't become overly fatigued. These motor units don't generate enough tension for movement, just enough tension to maintain tone of the muscle. And this you'll see decrease during deep sleep. It's one of the reasons why people snore when they're sleeping is because they lose the muscle tone in their throat muscles, essentially. And their soft palate can kind of hang lower, so to speak. Okay, isometric and isotonic contractions. Isometric contraction, this is going to be where the muscle contracts and muscle tension increases, but the muscle length overall is going to stay the same. So an example of this would be holding a weight while your arm doesn't move. You can have isotonic contraction. This is where the muscle tension is higher than the resistance, so this is going to result in movement. Muscle tone is going to remain the same, but muscle length changes, okay? So isotonic, muscle tone remains the same. Now, in the isotonic contraction, you can have either concentric or eccentric contraction, okay? Concentric contraction, this is going to be where the muscle shortens as it contracts. For example, if you were to lift up a load of laundry, your biceps brachii are going to contract and shorten as it contracts. Eccentric contraction is going to be where the muscle lengthens as it contracts. So, for example, when you go to lower that basket of laundry down on the ground, the biceps brachii are still contracting, but they're actually lengthening as you lower the load. Okay, here we have the difference between isometric and isotonic contraction. So isometric, the muscle tension is less than the resistance. Okay, so there's not going to be any movement that occurs. The muscle is not going to shorten, but tension is being generated. Here, the isotonic contraction, muscle tension is going to be greater than the resistance, so there's going to be movement. Whether the muscle shortens or lengthens is going to determine whether it's concentric movement or eccentric movement. Okay, so definitely know the differences between these two, concentric and eccentric, and isometric and isotonic. Okay, just a little bit about the effects of exercise in relation to the changes that occur in muscle. So endurance training is going to lead to better overall ATP production. You're going to see more mitochondria in the skeletal muscle cells. You're also going to see rich vascular supply to the muscles to make sure that there's enough oxygen getting to the tissues, and you're going to see increased myoglobin as well. Resistance exercise, this is going to lead to hypertrophy of the cells. So the muscle cells are going to increase in size. So this is going to be due to increases in myosin and actin. The muscle is also going to increase the glycogen reserves and the mitochondria as well. The changes from lack of exercise, this is going to be atrophy. So you see a decrease in muscle cell size just due to lack of use. This is initially reversible, but can become permanent if extreme. For example, someone who may no longer have the voluntary use of their legs may experience atrophy in their lower limbs. All right, what happens to muscle mass when we age? Well, we start losing muscle. Slow loss is going to begin around the mid-30s, and this is due to decrease in activity, metabolic activity. Okay, decrease, your metabolism starts to slow down in your mid-30s and just kind of continues on a gradual decline for the rest of your life. Something to look forward to. So you'll see a gradual loss of muscle mass if all other things remained constant, starting in your mid-30s. And you'll have a decreased size, power, and endurance of skeletal muscle, a loss in the fiber number and overall diameter. So you'll have decrease in those myofibrils, right, that make up the muscle cells. And you're going to have decreased oxygen storage capacity. Okay, cardiac muscle tissue. We've got cardiac muscle cells. These are short branching fibers. They're not the long tubular fibers of the skeletal muscle. Unlike skeletal muscle, which is multinucleate, cardiac muscle is going to have only 1 or 2 nuclei per cell. It is striated, so it does contain sarcomeres. Cardiac muscle has a lot of mitochondria. This is important for aerobic respiration, right? You don't want your heart muscles to get tired. Cardiac muscle cells have these things called intercalated discs that join the ends of neighboring cells. And these discs contain both desmosomes and gap junctions. And remember, these were ways that cells can sort of bind to one another. The desmosomes helps prevent shearing stress and helps keep the cells bound tightly together, while the gap junctions, in addition to binding the cells together, allows for the flow of cellular contents from one cell to another. So that's going to allow for fast transmission of the impulse of the cardiac cells to beat right, to contract and make the heart beat. Those contractions are initiated by the heart's autorhythmic pacemaker cells. So these cells that have their own rhythm, intrinsic rhythm, that beat on their own. Heart rate and contraction force is going to be influenced by the autonomic nervous system, and we'll get to that a little bit later in this course. All right, here's an image of a cardiac muscle cell or cardiomyocyte. Here are the intercalated discs. So this would be where one cell ends and joins another cell. Here you can see the nucleus of this cell. Here are desmosomes at one of the intercalated discs. Here are gap junctions at another intercalated disc. All right, so be familiar with all of these, and especially know the gap junctions. These, their location at the intercalated discs is important because it allows, like I said, signals from one cell to another to transmit very rapidly and allows the heart to beat in unison. Here you have cardiac muscle. You have the individual muscle cells here, highlighted in blue. You have the intercalated discs here, highlighted in blue. And then the nuclei here, highlighted in blue. Smooth muscle. Okay, so smooth muscle is found in a variety of organ systems with a variety of roles. Big picture here, smooth muscle is not going to be under voluntary control. This is going to be involuntary control, and it's going to be around things like your organs. That's where smooth muscles are going to be. In blood vessels of the cardiovascular system, this is going to help regulate blood pressure and blood flow. You'll find smooth muscle in the bronchioles of the respiratory system. This is going to control airflow in and out of your lungs. You'll find it in the intestines of the digestive system, for example, mixing and propelling materials through the small intestine. You'll find smooth muscle in the ureters of the urinary system. This is going to propel urine. You'll find it in the uterus to help in labor, help with contractions. There's the locations and functions are numerous, but big picture is this is not going to be under your voluntary control. This is all going to be happening automatically in the background, and it's not striated. Okay, we are not dealing with sarcomeres when we talk about smooth muscle. Smooth muscle cells have a fusiform shape. So they're wide in the middle with tapered ends, and you can kind of see that here, this fusiform shape. They are smaller than skeletal muscle fibers. The sarcolemma has varied types of calcium channels. So they've got calcium channels that's gated by chemicals, by voltage, etc. There are no transverse tubules in smooth muscle and very little sarcoplasmic reticulum. Instead, the most important source of calcium for smooth muscle is going to be the interstitial fluid, okay? And that's the fluid in between the cells. All right, so here we have smooth muscle cells. Note that they're fusiform in shape. You have these things called dense plaques. And the dense plaques are going to contain the dense body and intermediate filament and the thick filament and thin filament. So this is the unit, the contractile unit in a smooth muscle cell, not a sarcomere, okay? Cytoskeleton of smooth muscle cells is going to be composed of extensive intermediate filaments. We said dense bodies are going to be points where intermediate filaments interact within the sarcoplasm. And dense plaques are going to be points where the intermediate filaments attach on the inner sarcolemma. So here. the dense body, you can see the intermediate filaments attaching. The contractile proteins, again, are going to be actin and myosin. These are arranged between the dense bodies and the dense plaques. And contraction is going to cause a twisting motion, okay? We said like skeletal muscle, smooth muscle has actin and myosin that slide during contraction. Smooth muscle also requires calcium for initiation of contraction and requires ATP. But unlike skeletal muscle, smooth muscle has filaments that have latch bridge mechanism. This is going to be where the myosin attaches to actin for an extended period of time without burning extra ATP. Smooth muscle also has calmodulin. This is a protein that binds calcium to trigger contraction. And it has myosin light chain kinase and myosin light chain phosphatase. Myosin light chain kinase is an enzyme that phosphorylates the myosin heads when activated by calmodulin. And myosin light chain phosphatase is going to be the enzyme that dephosphorylates the myosin head. So myosin light chain kinase is basically going to be the enzyme that kind of turns on the contraction or initiates the contraction, whereas the enzyme that causes relaxation is going to be myosin light chain phosphatase. Okay, so here's smooth muscle contraction. Your first step is there's going to be some sort of stimulus that triggers the opening of voltage-gated calcium channels. Calcium from the interstitial fluid is going to enter the sarcoplasm. Calcium is then going to bind to calmodulin. You see calmodulin changes shape. Calmodulin is going to, the calmodulin with the calcium bound to it is going to activate myosin light chain kinase, myosin light chain kinase is now activated, and it goes on to phosphorylate the myosin heads, activating myosin. Now this takes a minute. Next, cross bridge formation, power stroke, and reattachment. Okay, so the activated myosin heads are going to bind to thin filaments to form cross bridges. Myosin ATPase breaks down ATP and gives that's where the energy for the power stroke comes from, okay, for the actual contraction. And then this process is repeated. The force generated is transferred to the anchoring filaments, and the smooth muscle cell shortens. So overall, big takeaways, it's going to be a slower process. It's kind of a more smooth, sustained contraction. It's not under voluntary control. The calcium comes from the interstitial fluid. It uses calmodulin, and it uses myosin light chain kinase. Okay, so smooth muscle relaxation. First, you're going to have cessation of the stimulus. Next is going to be removal of calcium from the sarcoplasm. Right, so it's going to get shipped back out into the interstitial fluid. You're going to have dephosphorylation of myosin by myosin light chain phosphatase. Everything's going to go back to its initial conformation, but this can take a minute to happen due to the latch bridge mechanism, right? Where we said myosin can bind to actin for an extended period of time without using extra ATP. That's the latch bridge mechanism. Okay, control of smooth muscle. This is going to be involuntary, or the autonomic nervous system. The muscle's response is going to depend on which neurotransmitter is being released. So some neurotransmitters are going to be excitatory. They're going to cause muscles to contract. Some neurotransmitters are going to be inhibitory. They're going to cause muscles to relax. okay? Just depends on what neurotransmitter is being presented and what receptors are there. Control of smooth muscle can also be stimulated by various hormones, low pH, low O2, high CO2, certain drugs. The big takeaway is it's going to be automatic processes, things that you don't have to think about consciously, okay? Okay, so multi-unit versus single unit smooth muscle. Multi-unit smooth muscle, what this is referring to is essentially if the smooth muscle cells move together as one unit or if they move independently as a multi-unit. Okay, so multi-unit, they're going to move independently. So they are arranged in units that receive stimulation to contract individually. So each muscle cell is receiving its own stimulation to contract individually. There's not going to be any gap junctions because you don't want these cells communicating with each other. You want them acting individually, doing their own thing. You don't want that impulse from the neuron that goes to this cell to travel to this cell, okay? This is going to allow for very fine control of the contractions, and you're going to see this in smooth muscles of the eye, the erector pili muscles, and the large airways of the respiratory system. So it's only in a few specific places. Contrast that with single unit smooth muscle. This is where cells are connected by gap junctions and are going to contract as a single unit. Most smooth muscle is single unit smooth muscle. You're going to find this in blood vessels, the walls of the digestive, urinary, and reproductive tracts, and in the smaller airways of the respiratory system. Varicosities. What does varicosity mean? Well, it basically just means swelling. So a varicosity is going to be a swelling of something. In this instance, we're talking about swellings of autonomic motor neurons that release acetylcholine or norepinephrine to stimulate smooth muscle contraction. Stimulus is going to spread from cell to cell through gap junctions. Remember, those gap junctions are really important for cell-to-cell communication and propagation of impulses. Okay, we talked about the multi-unit smooth muscle. Here's the single-unit smooth muscle. These smooth muscle cells are connected via gap junctions. So one impulse that gets sent to this muscle cell can travel to this muscle cell, okay? So they can communicate and share the input from the neuron with each other. Here's a close-up of the neuron, and you can see the varicosity here containing synaptic vesicles. And what's inside of those synaptic vesicles is going to be neurotransmitters. Okay, go over these questions and make sure that you understand these. And finally, here's a chart just to, for your own review to kind of give you an overview of everything we talked about regarding skeletal muscle, cardiac muscle, and smooth muscle. You've already become familiar with them as far as histology is concerned, so you should be able to identify them on a slide. So if I gave you a skeletal muscle cell, I would expect you to be able to tell me that that's striated muscle, and you would be able to tell me that it is skeletal muscle because the muscle cells are long and there's multiple nuclei there, all right? Here I would expect you to be able to tell me that this is cardiac muscle. I would expect you to be able to tell me this is smooth muscle. But on top of that, we're going to go a step further and talk about where these muscles are located, where their calcium source is located, what the contractile unit is. Remember that smooth muscles do not have sarcomeres. You need to know the stimulation. So skeletal muscle is going to be the nervous system voluntarily, whereas stimulation for the smooth muscle is going to be the nervous system, autonomic nervous system, right? The involuntary nervous system. And then the stimulation for cardiac cells is going to be autorhythmic due to the pacemaker cells. And then I want you to know, as far as the response and primary energy source, I want you to know this in regards to slow oxidative fibers versus fast glycolytic fibers, okay? And that you need to know for the test for sure. Again, this is a big chapter. There's a lot of physiology here. There's a lot of mechanics happening on a subcellular level, on a molecular level. So do not hesitate to reach out with me for any kind of clarifying questions. My office hours, I'm available to talk with you in person if that's what you need, or we can do a Zoom meeting, whatever you need. Just reach out to me. I'm available to answer your questions, okay? But this is going to finish up what's on exam number 3, which is tentatively scheduled for next week, and I'm going to send out an announcement about that here in a little bit.